So good morning, everyone. My name is Astrid Vicente, and I'll be moderating this last session of our conference. And I'm very happy to introduce to you the first speaker today, our first keynote speaker. It's Thorsten Gutjahr, and he's global head of companion diagnostics at AstraZeneca within the precision medicine and genomics function. And he leads the oncology team that has delivered 19 diagnostics and also plays a leadership role in AstraZeneca precision medicine strategy. So he'll be talking about precision medicine from the patient needs to providing solutions in clinical practice. Thank you very much.
Thank you very much, Astrid, and thanks for the organizing committee, not only for having me today, be able to present, but also actually for organizing this conference. I was really impressed, and it was a pleasure actually to listen to yesterday's presentations. I think they were very deep understanding and fantastic progress as we do in personalized or precision medicine. We heard a lot about precision medicine and the advances that we have done and in its reality in several indications, primarily actually in lung cancer, obviously, and we heard a lot of different examples from lung cancer yesterday. Now, it goes obviously beyond lung cancer, and just recently at October's European Society of Medical Oncology, we have seen this news, this time not in lung cancer, but in ovarian cancer, for patients with a BRCA mutation, where obviously a new therapy, olaparib, extended life for ovarian cancer patients that were, as it was described, substantial and unprecedented, and also an estimated 3 year of progression-free survival extension. So very significant progress here as well, and we know this is in lung cancer, this is in ovarian cancer, it's in breast cancer. And in general, cancer is obviously the main disease area where we made most of the progress so far as an industry. I mean, I say as an industry, I should also say that I'm not only speaking on behalf of AstraZeneca here, but also FPA and EBE on behalf of the pharmaceutical industries. So, but there's much more we need to do and we can do. The need for new medicines and the need for new solutions goes way beyond oncology, and we do actually have significant efforts and activities in other disease indications as well, such as in respiratory diseases, in cardiovascular diseases, and beyond those areas as well. What we want to see is really a significant, tangible improvement for patients, and ideally extending patient lives, and making these disease and devastating diseases hopefully a chronic disease in some time of the future. So the concept of precision medicine is a straightforward one. Identifying patients through, first of all, having access to the suitable high-quality patient samples, then doing the research, doing the translational sciences to identify the right biomarkers, the right patient segments, and then actually through the use and the development of a diagnostic test, identifying the right treatments for our patients, for these patients, with the aim to drive an overall benefit for patients, ideally extending of life of patients and high quality of life for patients. Now, the concept is simple. We know the implementation actually is not as simple. And from a development perspective, from a pharmaceutical development perspective, it actually means a different mindset, a different approach, how you develop actually medicines, how you work with the academics, with biotech companies to identify patients and the right treatment for these patients. And that has taken time, but we see now the fruits of these efforts that have actually started 5 or 10 years ago in clinical practice. So through these efforts and through this understanding how to actually now go into a targeted drug development, have the patient selection right to actually target it to the right patients that benefit most, what we have seen is there are more and more approved medicines with a companion or linked diagnostic, in clinical practice. And more of approved medicines actually come exactly with that combination, medicine and diagnostic, to identify those patients highly benefit. So that's the fruit that really have taken off 10 or so years ago, and significant progress have been done over the last 4 or 5 years. So a work has been commissioned, a study has been commissioned from FPI and EBE to actually look into this in more detail. And Charles Rivers Associates have done that work and actually published it just this year. And what they did is actually, through this exercise and through these studies, they classified the benefits of precision medicine and the promise of precision medicine in 3 categories. First, and most importantly, deliver better treatments for patients, extending patients' lives, overall survival, and have improved efficacy for those medicines and for the patients. Secondly, not only for patients, but the whole society and healthcare should actually benefit from that. Improvement of patient management, as well as prevention and delay of more expensive costs, allowing actually the healthcare resources be really focused most efficiently. And the third pillar was that they identified that actually we can have a more efficient development as well. As we know, development of medicines take a long time, years, decades to do, and can actually this be enhanced, more targeted, speeding up. So also here the resources and the efforts we're putting into this are used most efficiently, and there's reduction in costs as well. So the question was, actually, are we seeing progress towards these 3 pillars? First, and we heard this yesterday in several examples, we know that this approach and precision or personalized interventions have led to better patient outcomes. There was a study, for example, here in melanoma patients over actually only the past few years, where they looked at the 1-year survival for melanoma stage 4 patients, or metastatic patients, devastating disease, looking at did we actually make progress there. And as you can see, the 1-year survival over the years, year after year, actually extended through better care, but also through earlier and better and targeted intervention, new medicines being available for patients that really drive this 1-year survival benefit here. And we have many more examples, also what we have heard yesterday. Secondly, does the healthcare environment actually benefit from precision medicine as well? And there was an interesting study from INCA, from France, where they funded and allocated testing investments into each of our testing. And this resulted actually in a substantial increase of screening of patients. And with this, they estimate that would have saved, or that will save, about 70 million of the French healthcare insurances through better treatment and through different treatment, more targeted treatment for these patients, because they have been identified. And we heard a similar result actually from Germany yesterday as well, that they actually drove the same thing. So there's more and more proof that it benefits patients. It also benefits other major stakeholders and the healthcare system societies as we go and use these approaches. Third, does actually precision medicine help the drug development? Can we be more successful? Can we be more targeted and potentially faster? And a study has been looked at to investigate this. What is the success rate for medicines that have developed through a precision medicine approach versus non-precision medicine approach or non-biomarker approach? And what you can see here on the last pillar, actually, those that come actually with a precision medicine or biomarker-driven approach are much more successful from phase 1 to approval than those that come without. This is fantastic news, and it's good progress. But again, we need to do more because we want to drive this 25% or 26% up to a much higher rate, so that we can actually offer these solutions to patients and to the clinical practice, more often and much more efficiently. I want to change gears now a little bit and move into some case studies, go a little bit into the science on a high level, and see what progress have been made so far recently. The first one is actually that has been mentioned already in several talks yesterday and presentations is osimertinib. It's a novel medicine for EGFR mutation positive non-small cell lung cancer patients. I think let's go back actually to what we have learned through the last decade about lung cancer and the specific lung cancer where we detect an EGFR mutation. So we know now for quite some time that certain mutations in EGFR leads to an oncogenic phenotype, and that drives the tumor. And we do have, drugs available, such as gefitinib or erlotinib, or so-called first-generation tyrosine kinase inhibitors that are targeted to exactly those patients. And they're really a profound benefit to these patients into extending lives, extending progression-free survival, but also overall survival. Now, we also know that patients on these treatments actually progress, unfortunately. So they develop a kind of a resistance to those agents, and the tumor will spread up again and grow. So investigations and research, and we do put a lot of research into this with our partners in academics and biotechs to understand these resistance mechanisms. Now what we have learned here is that actually a pretty large fraction of these patients on a first generation tyrosine kinase inhibitor actually progress due to another mutation, and it's about 50 to 60% of those patients develop the so-called T790M mutation, another mutation within the same binding pocket in the EGFR protein or gene. And that's a gatekeeper mutation. So what has been done through this understanding and through this biology, our researchers and actually chemists now went into this and looked at this and said, how do we define and design a molecule that inhibits, on the one hand, these activated mutations, L858R and exon 19 deletions, which was the traditional sensitizing mutations, but also the T790M mutation. Can we design a molecule that is optimized to bind exactly this and actually inhibit all three mutations? And that's basically the birth of this medicine. This exactly has been done. It was a designed molecule to exactly do this based on the biology, based on our scientific understanding. This has been a very quick, rapid development actually. And this molecule has, great characteristics, so we could go in early clinical trials in phase 1. This is a patient actually, non-small cell lung cancer patient, and you can see the tumor here on this scans with an exon 19 deletion and the T790M mutation after gefitinib. So the patient progressed, and that was actually in the lowest dose in the phase 1, so a dose escalation. So it was the lowest dose for this new medicine, osimertinib. And even on the lowest dose, you could see that effect. So we knew there is something active. We have a drug in hand. So let's, speed up the development, because it was so encouraging to see this, even if it was the lowest dose. And then with dose escalation, we would expect an even better benefit for more patients. And that was the result of the phase 1 trial. You see the waterfall graph. When the bars go down, this means the tumor shrinks. And you can see that most of the patients actually benefited in this group of T790M positive mutated patients after first-line tyrosine kinase inhibitors and progressing on those. So that was extremely encouraging, and of course it went on, and now osimertinib is actually available for those patients in clinical practice. Now, what also became apparent was that, unfortunately, this is after first-line therapy, so sometimes for many patients, it's very difficult and almost impossible to get actually a tumor biopsy from these patients. The tumor biopsy may not be suitable, or actually somewhere where we can get a suitable biopsy. So what solutions do we have for these patients? Because we cannot test them for this new so-called resistance mutation. And so research went on in the whole community and with academics. And we know now that obviously in cancers, in tumors, that through different mechanisms, active and passive mechanisms, as there's apoptosis and necrosis and also active secretion happening, that tumor DNA from the tumor cells actually are released into the bloodstream. And so obviously in the context of a lot of DNA in the bloodstream that is from non-tumor cells. So what technologies do we have available to detect those in the bloodstream, so-called circulating tumor DNA, to detect these same mutations and actually then have an option available for these patients? So a lot of research went on. You could use this technology and this new opportunity now to either look at single genes, such as EGFR TP90M. You can think about, we need actually a panel of molecules and genes to detect and do a targeted gene panel. Or even, we're now going into do it in whole genome. And this is still research ongoing and development ongoing, but eventually we will get there. And the sensitivity is increasing gradually as new technologies and as we learn more. And also the specificity is extremely high. And so it's really important for these patients to also be detected. Again, now for T90M, this is actually now available. So we have tests available to look at tumor tissue. And if tumor tissue is not available, we have an opportunity now and use technologies in clinical practice to look at plasma DNA and circulating tumor DNA as well. So obviously, the research and the clinical studies went on, and we then moved from a second line into a first line and said, if osimertinib is so active, how does this look like against the traditional first line TKI standard of care medicines and medication? And so that was the so-called FLORA trial, and you can see on your left-hand side the results, osimertinib against standard of care, so first-line tyrosine kinase inhibitors, with a very significant, clinically meaningful benefit for osimertinib. Now, again, the same question was true. This looks at now when we detect them by tumor tissue, what about plasma? And so we developed a plasma tests with our diagnostic partners and looked at retrospectively, does plasma, what results does give us plasma, and this is what you see on your right-hand side. Visually the same results basically, and again, with further research being done and development, this is now also available. So we have different solutions and different modalities we can detect these and identify these patients that benefit, in this case, as upfront therapy for patients with an EGFR mutation. Second example, I want to go into the PARP inhibitor olaparib for patients with ovarian and for breast cancer, and research is ongoing with other cancers as well. So let me go first actually to explain how PARP inhibitors actually work. Single-strand breaks in our DNA happens every day in all our cells, and this is what kind of biology does basically. But that's not a problem, because we have an enzyme that repairs the single-strand breaks. Now, what if we inhibit this? We inhibit the single-strand breaks repair mechanism. What happens from a single-strand break, double-strand break actually happens. They're more serious actually for DNA damage. Now, even that is not such an issue, because we have several mechanisms, of which the most important one is homologous recombination repair, in our cells that repairs double-strand breaks. Great biology, it works. Now, what happens if this is not in a normal cell, but in a cell where the second mechanism of repair mechanism, homologous recombination repair, is inhibited, is not working? And we have these cells, and there are these tumor cells and patients that suffer from this. And these are cells with so-called homologous recombination repair deficient tumors. And for example, and we know it well, the gene, the BRCA1 and 2 genes, there are these mutations that inhibit homologous recombination repair. So in these patients, this mechanism is not there anymore. So think about, if we have now a molecule that inhibits the single-strand breaks, the PARP inhibitors, so the double-strand breaks happens, and in the context of a BRCA mutations or other mutations that inhibit homologous recombination repair, what happens to these tumor cells is they accumulate DNA damage more and more and eventually go in cell death. And that's the theory, that's the research, and to be honest, it's much more complicated than I just explained. But that basically was the research done on olaparib or on PARP inhibitors in general. Lots of research being done in clinical studies with the outcome of the latest phase 3 trials, on the one hand in ovarian cancer patients, and on the other hand in breast cancer patients, with a very significant and profound improvement of progression-free survival in both diseases with BRCA mutation, which is extremely promising, and the SOLAR-1 is actually the press release that I showed earlier from ESMO this year. So very significant and tangible improvements and significant improvements for these patients, and hope for these patients to actually have the right thing if we identify the right patients with BRCA mutations or with other mutations that inhibited homologous recombination repair. And this research is as ongoing, and we have now a phase 3 trial, for example, in prostate cancer patients, where we look at a broader panel of mutations that inhibit homologous recombination repair, and we are eagerly awaiting the results to really refine our approach to identify those patients. So far, so good, extremely promising and good efforts done so far, but how can we further improve this? I mean, as we go further, where are the elements that we need to look at? First of all, regulatory systems are extremely important as we develop solutions for medicines and for companion diagnostics. How are these regulated? What opportunities do we have? How can we ensure we do further research as we go through the clinical trials and in also clinical practice? And so we are discussing on a daily basis, basically, with our regulatory colleagues and regulators around the world, what systems can we put in place to protect the patient, to ensure that we do the right thing for patients, but at the same time, it's a pragmatic approach to research, and we can get these solutions quickly to patients. Reimbursement has been a big debate yesterday as well. We had a fantastic presentation yesterday morning about reimbursement situation, reimbursement environment, to get, making sure that the test and also then the precision medicine and the medicine is actually reimbursed and getting to patients. Biology, and it was mentioned yesterday, this is the fundamental. Without understanding of biology, without understanding of the science, enabling our scientists to do the best work, collaborating around the world is critical. Development of the technologies from a research side of things, but also from a development and diagnostic is absolutely critical. So further investment needs to go into this new technologies investigations. Having a coordinated healthcare delivery system, continuously educate health practitioners, ourselves, the whole community, and I think this here is a wonderful forum where exactly we do that, to kind of share our experiences and share what we can do better. And mechanisms of sharing, and we heard the thing about data and algorithms that we develop, and this is all only possible if we share those in a way where it protects the patients, and then the other way we can actually do our research together as a community to advance our search for better and good solutions in precision medicine. So again, the research that have been done through FPI and EBE actually identified then 5 priorities for precision medicine in Europe. The first outline is actually to have prioritization on a national policy level for precision medicine. The secondly, of a streamlined care management and resources, driving consistency and making sure that there's consistency, that all patients can benefit from these solutions. Diagnostic investments and funding on new technologies, on new diagnostic solutions, and funding pathways for doing this and diagnostics. Data and quality, on the one hand data sharing, on the other hand also that we make sure that the data we get from these tests are accurate. So external quality assurance programs are absolutely essential to make sure that labs, laboratories around the world can actually have an assessment how good they are doing and what to potentially further improve, because it's a decision for patients, so we need to make sure that test results are accurate and robust, and access then to the medicine, reimbursement of for new treatments, and aligning requirements for health technology methodologies. We heard yesterday morning that some of the assessments are not suitable for precision medicines and diagnostics, clearly from Catherine yesterday. So again, so what else and what do we need to change to actually adapt those to those solutions? So the report also outlines the barriers and the enablers. I'm not going through all of this, it's a busy slide. But importantly is, testing, get good testing, that patients get tested for those solutions that we have already out there, which includes, treatment guidelines are extremely important. That drives testing, early access schemes, and also then reimbursement of the diagnostic and the medicine, so patients around the world can benefit from those solutions and gain that benefit for improved progression-free or overall survival. So this is my last slide. I think we came a long way. I talked about the BRCA story and PARP inhibitors, and BRCA is an extremely complex gene. It's actually 2 genes, BRCA1 and 2. Actually, you have variants of over 10,000 variants in BRCA. About 4,000 of them are actually those that need to target where homologous recombinant repair is not working. So this research is ongoing, but we found a solution to overcome these hurdles, and also with regulators together, we collaborate with them to find a solution to get the diagnostic approved and the medicine then available to patients to identify those patients. Circulating tumor DNA is now a reality as well, and I said it's now going into panels and maybe whole genome analysis for ctDNA. So here we have solutions already on clinical practice, but we need to keep on going and keep on doing, because we can do even better. At present, our research across the industry, across academic centers, are going into immunology, which is an area I didn't touch upon, but it's extremely important to patients and extremely important to all of us. It has a different mechanism, obviously. How can we identify those patients? What are the diagnostic solutions we can provide and do research and provide it then to physicians and patients in clinical practice? Multi-gene DNA panels is common practice now in development and research, and coming more and more actually into clinical practice as well. And also point of care diagnostic solutions, where obviously sometimes we need solutions quick. Patients cannot wait, physicians cannot wait for their test results. When we do an NGS, a next generation sequencing based test, it may take 10 days, maybe 2 weeks, maybe 3 weeks until we have a test result. For some lung cancer patients, but also for some patients maybe with cardiovascular complications, this is not the time that they can spend. So how can we develop further solutions to get a test closer to the patient and faster as test results, so we can have the right treatment for them available? And the future is actually what we do now in our research and development area, but that could and will become actually sooner, I think, reality than we all think. How quickly the development goes into how we look at genomics, and we had several very great examples from Sweden yesterday on their genomics initiatives. Early diagnosis and monitoring is actually key for patients, the holy grail basically to detect tumors and disease as early as possible, so intervention. So the benefit that patients get from this will be much more profound. Sensors, so continuous monitoring on health, and a lot of efforts going in there, especially in cardiovascular, metabolic diseases, and the use of machine learning, artificial intelligence, to actually benefit from the data we are generating for the best benefit of patients. So if this, hope I gave you a good overview, and thanks a lot for listening. Happy to take any questions.
Thank you so much, Thorsten, for a very interesting talk. I wonder if there's one question. We have time maybe for one question.
Very impressive data. Harald Schmidt from Maastricht. I'm still surprised that you can use organ-based tumor definitions. I mean, one of the main outcomes of the Pan Cancer Atlas was that basically we should no longer use organ-based tumor definitions, but convert them into mechanism-based definitions, usually pathways and not single targets.
Absolutely. I think that's obviously what's happening in various areas. Sometimes mechanisms, when we see different mutations, present differently in different diseases. So although it's very logical, we still need to test, does the mechanism still hold true, and do we see the same benefit for patients? But you're absolutely right. That's going to be, a major effort, and is already ongoing, as we go tumor agnostic, as we call them, and actually go through the mechanism to make sure that, we're not talking about lung cancer, breast cancer, etc. anymore. So, but we do need to do the research and the results, and then getting this. But I agree with you. That's going to be a major. It's already in major development, and a solution that we hopefully will bring to patients as quickly as possible. It's a good point. Thanks. Thank you.